Hi, everyone. Today we are continuing on with chapter 3. And now that we've talked about all the structures, we're going to talk about some of the functions that the plasma membrane plays in the cell. So the membrane, as we know, separates the outside of the cell from the inside of the cell. And the phospholipids are able to regulate the movement of things coming into the cell and going out of the cell. So there are two terms here called passive and active transport. And these are different from each other in that one will require energy, that is active transport, and one does not. It will depend on other things to drive the movement of stuff. So we're going to start off with passive transport, and this is the transport of stuff, whether we're talking about ions or nonpolar molecules or whatever, going from one side of the membrane to the other. And passive transport is not going to depend on any sort of energy. So one type of passive transport is known as diffusion. And diffusion is just the movement of a solute from a high concentration where there's a lot to a concentration that is lower. So what this movement will do is it is going to balance out the concentrations on both sides of the membrane so that both sides have the same ratio of solute to solvent, which would be the water. Okay, so if you have a lot on one side, there's going to be pressure for those particles to move to the other side where there's less. All right, you can think of it like our classroom, and normally we would have an example where there would be a lot of you on one side of the room, and then we would demonstrate how it's kind of uncomfortable, cramped, and not a whole lot of fun to be all crammed together in one little corner of the room, and you'd rather be on the other side where there are less people, more room to spread out. So diffusion is similar to that in that the solute, where there is a lot, they want to move to where there is less. Okay. So the difference in concentrations on either side of the membrane is known as a concentration gradient. And these things, the solute, they will move down the concentration gradient from a high concentration to a low concentration. All right. So because of the force that is there, okay, kind of pushing the solute from a high concentration to low concentration, no energy is required here. Another type of passive transport is facilitated diffusion, and this is the same idea as the last one, but instead of being able just to pass through the phospholipids here, there are charged particles that are going to need the help of a protein to go from one side to the other. So it still works going from a higher concentration to a lower concentration, but instead of just passing directly through the phospholipids, there needs to be a helper protein there to bring the particle from one side to the other. Okay, so things like ions, glucose sugars, amino acids, things that have polarity need the protein to help it to go from one side to the other. Okay, but like I said, it's still down the concentration gradient, going from a high concentration right here to lower concentration, so the movement would be into the cell in this case. Osmosis. So I'm sure some of you have done a lab where you've taken an egg, dissolved away the shell, and then manipulated the egg by putting it in different solutions. So if you put it in water, the egg is going to swell up. If you put it in corn syrup, the egg is going to shrink and shrivel up. And that's because water is moving through the membrane on the outside of the egg. So if it's put into water, water does not have a whole lot of solute. So the movement of the water is going to go into the egg or into the cell where there is less solute. Or I'm sorry, where there is more solute. Right? The goal, again, is to balance out the concentrations on either side. So the water is going to move where there is lower concentration to where there is higher concentration. So let's say that the egg is put into the corn syrup where there's lots of sugars. Well, the water is going to go from inside the cell out into the corn syrup because of osmosis. Once again, because the inside of the egg has less solute than the outside of the egg. So this is maybe a little bit confusing. Unlike diffusion, the water is going from where there's a lower concentration. But remember, the water is not the solute. In the previous examples, we're talking about salts or sugars or other things that can pass through the membrane. But in this case, the water is the only thing that can move through the membrane. So the water is going to go from a lower concentration to higher concentration because, once again, it is aiming to balance the concentrations on both sides of the membrane. So that brings us to three terms for solutions in which a cell or egg or whatever can be put into that will determine the movement of the water through osmosis. So if it's an isotonic solution, iso means equal, so on either side the concentration is going to be equal, so there's going to be no overall movement of water. A hypotonic solution is a solution that has a lower solute concentration, so its osmotic pressure is going to be lower, meaning that the water is going to flow into the cell, and as a result, the cell will expand because of the added water. In a hypertonic solution, it is the opposite of the hypotonic. So now we're in a solution that has a higher solute concentration, and it is going to pull water from the cell. So water will flow out of the cell, and as a result, the cell will shrink or look shriveled. So a nice little diagram here. So we're looking at red blood cells in different solutions. So in a hypotonic solution, that means that there's less solute outside of the cells, so water will flow into the cell to try to balance the concentrations. So the cell will swell up, but what can happen in a hypotonic solution is the cell can swell up so much that it actually bursts, which is bad. Hypertonic is the opposite, so now there's a lot more solute on the outside, so water will flow out of the cell, which will shrink and shrivel it. In isotonic solution, there is still movement into and out of the cell, but now it's balanced. Okay, rather than a net movement of into the cell or net movement of out of the cell, there is still movement in and out of the cell. However, it's balanced. So over time, the cell is not going to change in size. That brings us to filtration, which is related to the Strawberry DNA lab. So with filtration, molecules will actually be forced through the membranes. This could be water, as is the case in blood vessels, or it could be other solutes. So with filtration, blood pressure is an example where these things like water or like dissolved substances like ions will be forced through the thin walls of a capillary because of the presence of proteins. So it's kind of like osmosis, right? The proteins are inside, they're forcing the water and these other things out. So, there's a little bit more force behind it here. So blood pressure is very important for getting things to cells that need the water or need the ions or need the nutrients. Okay. So if you ever have inflammation, inflammation is your body trying to heal an injury or fight off an infection. And one of the characteristics of inflammation is the presence of additional fluid to help to heal the area. And that's a condition known as edema, where the fluid and the other stuff, the nutrients, will get sent to the tissue to help to heal it and fix that injury. All right, and that brings us to active transport, where now we need energy. Okay, so sometimes energy is needed for the function of a carrier protein to send things from one side to the other. But in all of these other ones, endocytosis, where we have pino, phago, and receptor-mediated cytosis, all three of these are examples of bringing things into the cell, basically pinching off part of the membrane to form a vesicle to bring the stuff inside of the cell. So endocytosis includes all the other ones that we saw. So the molecules are going to get engulfed by the membrane, and it's going to pinch off and be brought into the cell as a vesicle. Okay, so remember, the vesicles are surrounded by membrane, and the membrane comes from the plasma membrane in endocytosis. Okay. So one example of endocytosis is cell drinking, aka pinocytosis. And all this is your cell is trying to bring in fluid, and it does so by engulfing some of that extracellular fluid with some of the solute inside, and will pinch off and bring it into the cell in a vesicle. Phagocytosis is cell eating, where larger things are going to be engulfed, whether it is larger molecules like maybe a protein, or perhaps even a bacteria for like a phagocyte that would be eating it to destroy it. So these larger molecules are brought into the cell. And again, an example are phagocytes, which are types of white blood cells that will actually help to clean up the extracellular fluid, which may have pathogens like bacteria that need to be taken care of, and they will do that, and they'll destroy them. But first, they have to phagocytize them to bring them into the cell. All right, but they do the same thing with cellular debris. If there is an old structure that is not working anymore, okay, it can break off a piece of it and then help to break it down, and then now you have more parts to build something new with. And the last one is known as receptor-mediated endocytosis. So this is a little bit more specific in that there are receptors on the outside of the cell which will accept these ligands, and they will specifically bind just to these molecules. And when they bind to them, right, that will initiate the process of pinching off part of that membrane to bring them inside of the cell. Okay, so an example of receptor-mediated endocytosis involves low-density lipoproteins, where molecules like cholesterol are packaged and brought into the cell. Now, the opposite of endocytosis is exocytosis, and molecules, instead of coming in, are being sent out. So remember, the Golgi apparatus is packaging up proteins and other large protein-related molecules and making vesicles and then sending them to other parts of the cell. But sometimes the vesicles are going to want to dump the contents outside of the cell so that those proteins or whatever can get sent elsewhere. Okay, so the vesicle is going to travel from the Golgi apparatus to the membrane, and then it will attach to the membrane, dump the contents out, and now the membrane that was part of the vesicle is now part of the plasma membrane of the cell. Okay, so again, the secretion of proteins utilizes this process where the vesicles are pinching off of the Golgi apparatus, and then They travel to the membrane and then deposit the proteins outside the cell and become part of the membrane. Okay. So, what is this? Chapter 9, I think, we'll be talking about the nervous system and this. So, vesicles and exocytosis is how nerves release their little chemical signals that go from one nerve to another nerve or perhaps to an effector. They'll release these neurotransmitters in that way. So that is all for today. Go ahead and complete the assignment and then have a great weekend.